Welcome everyone, and thank you for joining us for today's webinar, Advances in Three-Dimensional Cell Culture in Drug Research and Discovery, presented by Dr. Richard Eglen, Vice President and General Manager of Corning Life Sciences. My name is Dr. Susie Valdez, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Just simply click on that Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, just simply click on the Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of your presentation window and follow the process to obtain those credits. Now, without further ado, please join me in welcoming our presenter today, Dr. Richard Eglen. I will now turn the presentation over to him. Welcome, sir.
Thank you, Susie. It's a pleasure to talk to everyone today about this exciting field of 3D cell culture, and as the title indicates, I'd like to talk about how it's got applications both for drug discovery research as well as developing novel disease models, and we'll talk a bit about that as well. The areas I plan to cover is really to give everyone a broad overview of this area of cell culture and how it is applied to studying cells in 3 dimensions. We'll refer to this as 3D cell culture. I'll then give an overview of the various techniques and tools used in 3D culture, and then spend the majority of the talk subsequently talking about all of the various applications of 3D culture as well. So the point being, if you go back and look at the history of cell culture, and you can stretch your mind back way back to a couple of centuries ago, it was recognized that embryonic cells from animals such as chickens could be kept alive several days outside of the body in a salt solution. And that then led to various methods by which one could study tissues outside of the body. It led in the early 1900s to the development of synthetic medias that were obviously man-made but optimized to keep different sorts of cells around. And then this also then led in the 50s to the development of microtiter plates, which have become the workhorse in many labs for both drug research and drug discovery. Of course, penicillin was developed in the 20s. That enabled the foundations to be laid for having effective cell culture methods free of contamination from environmental bacteria. And then the first immortalized cell lines were developed, the famous one being the HeLa cells from Henrietta Lacks. That was one of the first immortalized cell lines that were developed for widespread drug research. Various improvements came along in the 50s, including the ability to coat glass surfaces and indeed other surfaces with proteins such as collagen that improve cell attachment and growth. And then plastic came in as workhorses in terms of growing cells, flasks, dishes, 96-well plates as well. Anchorage-dependent cells were being developed at that time such that you could grow them on other sorts of surfaces, including microcarriers, very, very small particles, which increases the surface area for growth. And then on the cell culture side, there was a development of fused cell hybrids, different sorts of cell lines fused together, and the creation of hybridoma lines, which were used initially for the production of various antibodies. So all of that activity led up to the late 70s, whereby the routine culture of cell culture was normalized, where the routine growth of immortalized cell lines was widely established, and the development of synthetic media and surfaces by which you can optimize your conditions. Going from the 80s onwards, it became apparent that you could maybe improve this from growing cells on a flat surface in a two-dimensional system to starting to look at cells as to how they appear in three dimensions. In other words, mimicking the situation of the cells in vivo. And artificial 3D matrices were developed in the 80s, and then a milestone was reached in 1987 by the development of Matrigel, which is now available through Corning Life Sciences, and that was first used to grow mouse sarcoma cell lines. In the 80s, human embryonic cell lines were first isolated. In the 90s, implanted organs were first grown from lab-grown sources. And then there was a development of a range of novel surfaces, including the development of ultra-low attachment surfaces on a whole variety of vessels. And these enable different cell lines to be grown. Once they'd adhered, they could be expanded, and they could be grown in very large vessels indeed. Bioprinting started to enter the field in the 2000s there, where you could now print synthetic tissues as well as just letting them grow, and that started to open up the ability to have architecturally organized, spatially located cells, and that has led in turn to the development of organoids, including, for example, in 2008, the 3D culture of brain tissues, where you're now starting to recapitulate not just cells grown in 3D, but cells that are grown in the right location, in the right proximity to other cells. And then this field has developed such that by late as 2009, for example, complex structures such as blood vessels were beginning to be bioprinted as well. So I put this in because I thought the audience might be interested in putting 3D cell culture in sharp perspective, that it represents the end of a long line of development of technologies by which you could take cells from the body, grow them outside of the body, look at them as disease models, look at them as mechanisms to interrogate for drug action, and 3D culture represents the culmination of many of these years of activity. In fact, if you look at this slide here, there are basically three types of cell lines used in cell culture. The first one, of course, is, and I refer to this with the HeLa cells, is immortalized cells. These were originally taken in general from animals and humans. They are optimized to grow on plastic surfaces, and they are indeed used widely throughout R&D, both in the academic setting as well as in the drug discovery setting. They are easy to grow. They grow in a very reproducible fashion in very large amounts, and for that reason they have become the workhorses for many R&D labs. The other major cell line type are primary cells, which are taken directly from the human body. They're often grown on biological surfaces, and they are increasing in terms of their use in terms of drug discovery. Major reason, of course, is that they can be taken directly from the patient, and as a result of that, they have the ability to carry many of the disease characteristics on board, such that you can start to look at drug action there. And then finally, stem cells have started to become more and more routinely used. The advantage of stem cells is that you can isolate them from several human tissues, you can grow them on biological surfaces, and they don't simply provide optimal disease models, but they can themselves be used as therapeutics. The point being here is that if you look at the continuum from left to right, the relevance of these cells goes from low to high, in that immortalized cells, by virtue of being immortalized, tend to have a low relevance in terms of biology, but stem cells, of course, have a very high relevance. Contrast that with the difficulty of using them. Immortalized cells are easy to use, easy to grow, easy to isolate, but as you go through primary cells and indeed stem cells, they become more difficult to work with, more difficult to isolate, and as a result, more difficult to use. Now let's focus now on three-dimensional cell culture, because the goal here and the theme I'm developing is that one wanting to look at cell culture techniques and cells themselves where they have improved biological relevance. That's important in disease modeling. It's very important in terms of looking at the pharmacological action of drugs. And growing cells in 3D culture enables one to take a step in that direction. The slide here shows, in fact, what happens to cells, and the figure on the left there, the 2D cell culture, is a cell grown on a plastic plate. There's some sort of surface treatment there, but you can see that in contrast to the cells grown on the right, where they have spontaneously come together to form an aggregate, a three-dimensional structure known as a spheroid. And two things to note, one of which is the appearance of the cells looks very, very different, but of course the figure on the right starts to mimic how cells interact in the body. And in fact, if the structure on the right was a model of a tumor, for example, then these spheroid structures mimic very well, and many of the cellular associations one sees in cancer there as well. Parenthetically, it's not just the appearance of the cells that is different, it's also their genetics, it's also their phenotype, and it's often the expression of cell surface proteins that differs from when you look at them in 2 dimensions as to when you look at them in 3 dimensions. Now this slide here shows, in fact, the spectrum going from 2D, illustrated on the previous slide, right the way up through to increasing levels of complexity. So taking the, going from left to right here, you can see that 2D culture, as I alluded to on the previous slide, can now be taking those cells, growing them in terms of a spheroid, where they form spherical aggregates there. If you leave them to differentiate, or you add various growth factors or differentiating factors or specialized media, the cells can differentiate into organoids, where you tend to get aggregation of more than one cell type, and you start to set up various primitive organ-type structures. Of course, these can also then continue to develop into tissue systems, in some cases all the way through to simple organs. Illustrated here is the skin, for example, right the way up through on the far right there, complex organ structures, illustrated here where we've got an assembly of cells forming a liver system. And of course, that itself is very important because now one can start to look at the effects of compounds on the liver outside of the body. And I'm going to come back to that at the end of this talk, as that's an illustration of the power of 3D cell culture. And similar to the previous slide, 2D, 2 complex organ systems by way of 3D culture, of course, goes from low relevance to high biological relevance, but the complexity increases as well, as you would expect. So low complexity in 2D, high complexity in organoids and complex organ systems. Taking a spheroid here, and this biochemical illustration shown on the right of the slide is meant to indicate some of the intricate microenvironment one can now start to detect when cells come together in assemblies. This is a spheroid again, and you can see here with the various sections, studied with a whole range of different technologies, how the cells can be studied in terms of a 3D structure. And by the way, throughout the talk, where I've used illustrations from the literature, the primary literature reference is also indicated on the slide here, and subsequent slides, as you can see. Taking the image on the right, you can see with a spheroid, for example, it's well known that as cells assembly into these aggregates, the oxygen gradient decreases from the exterior to the interior, and that's shown there on the upper right of the diagram there. And as a result of that, you get correspondingly subsequent biochemical changes. The distribution of ATP, reflective of metabolism of the cells, varies. So on the outside it's high, on the interior it's low, and that is also reflected in the distribution of glucose there. You can see it's high on the outside, it's low on the interior as well. Correspondingly, since low oxygen conditions lead to lactic acid accumulation, you can see in the center of the spheroid, lactic acid accumulation is high, it's lower on the outside. And indeed, all of these effects biochemically collectively relate to, in some cases, differences in differentiation and proliferation of the cells. And in the interior of the spheroid, you can, in fact, after time, get frank necrosis. And that's illustrated not just by the histopathology, but right down at the DNA level, where you start to see DNA strand breaks, which is themselves a reflection of necrosis or cell death, including apoptosis. Now, this is what one can see in a spheroid, and of course, this is exactly what one sees in a tumor. And as a result of this, in this case, if you had the appropriate cell, you would have a very good model, in fact, in a 3D cell culture system of what the tumor would look like in the body. When one wants to study spheroids, you can now do this routinely. And in this case, Corning is one of many companies making microplates, although Corning is the only company that can do this in an ultra-high throughput screening system using a 1536 well. Spheroids will grow very well in microplates. And you can see that if they're in a 96 or a 384 or a 1536 well format, that can be used for both drug discovery and drug development. And increasingly, spheroids are used in high-throughput screening campaigns, but they are also increasingly used as toxicology models, whereby you can get good prediction of the effect of compounds in terms of their potential drug toxicity or drug metabolism. Not to be covered today is the applications of spheroids and 3D culture in other areas. And this field is moving into bioproduction, particularly in the production of vaccines as well as biologics. And finally, with mesenchymal stem cells, growing them in 3D, particularly generations of embryoid bodies, they can be used for not just therapy, but also areas of tissue engineering. Today, I want to focus on the application of 3D culture, spheroid culture, in the use of drug discovery and drug development. The continuation of spheroid, as I mentioned on one of the illustrations previously, is how 3D culture is now moving into tissue organoids. And these are providing near-physiological models by which one can start to look at the development of human cells, human organs, as well as the impact and development of human disease. And in fact, you can consider that an organoid is a variety of tissue culture techniques that can provide a system of cells that is self-organizing, self-differentiating, and self-renewing, giving you a 3D cell culture system. Now, some examples of this are shown coming up here. For example, if one takes a pancreatic organoid, and these assemblies of pancreatic cells which have differentiated are grown in a matrigel, then you can see with these mouse cells here, the goal of this experiment was to study beta cells in the pancreas that can produce insulin, and how can you modulate these cells and optimize them as means to produce insulin, and therefore study some of the fundamental disease processes involved in diabetes. Other cell lines that have been developed from stem cells to make complex organoids include this illustration here with lung organoids. Here the group was using cells to mimic the lung. They could produce definitive meso and pulmonary endoderms, and eventually they could produce branching airways and alveoli. And in fact, they use this as a disease model by which they could look at infections, for example, of syncytial virus. So growing the cells into a lung organoid also enabled them to start to develop a disease model and in fact start to look at how viral infection of lung tissue proceeds. And then a final example of this, of 3D culture when looking at organoids, this is a gastric organoid here as well. In this case, this is also grown in matrigel, just as it is in the previous slide here. Human iPSC cells have been grown into an endoderm tissue. The spheroids were then directed using various growth factors to form tissues that had gastric glands, pit-like glandular domains, and from this system, the researchers could identify novel signaling domains. In other words, looking at the fundamental organization and the biochemistry of the cells. And they could also look at infection of things like H. pylori as well, which is a fundamental cause of human ulceration. The slide here shows taking other intestinal organoids, looking at in 96 wells. That's the procedure we've developed here at Corning to develop intestinal organoids. It's done in a spheroid plate in a matrigel system, and you can see that in the illustrations at the bottom there indicate that you can start to see the correct cellular organization with the correct staining, allowing you to see a variety of different cell types there. So given these examples and given that long history I talked about and given the acceleration in terms of technologies, you could reasonably ask the question, if cell culture has been around for over a century, why now is the field only transitioning to study cells in 3 dimensions? And the answers are fourfold, one of which is that there are new advanced tools to make this easy and simple and reproducible to do. And in fact, shown there is a Corning ultra-low attachment spheroid plate, which makes it very straightforward to develop spheroid cell culture technique. But accompanied with this has been enhancements of the instrumentation. You can do better imaging. Shown here is looking at it down a microscope, but of course there are automated high-throughput imaging technologies widely available now, and that relates to the other side of it, shown on the third panel there, which is standard high-throughput instrumentation robotics have been applied to the study of 3D cell culture. And that's also accelerating it as a means by which it can be used in compound screening and compound drug discovery. And there's increasing benefits, of course, in terms of developing the cells in 3D for the use in terms of research as well as therapeutic benefit. Now, in the last slide, I do just want to just talk a little bit about some of the various techniques used. The slide here shows the major techniques by which cells are studied in 3D. One of the early methods of development was a hanging drop technique off a cover slip over a well, and in that drop of fluid, the cells assemble into a spheroid. I've also talked about the low attachment plates, whereby they're geometrically designed such that the cells will grow into a spheroid. You can look at them by inducing them into the fluid layer by introducing magnetic levitation. The cells have a magnetic particle on board. They will come together under the influence of a magnet. But increasingly, there are other techniques coming in. The use of microfluidics, the use of 3D bioprinting, and also the influence of growing cells on biological hydrogels as well as synthetic hydrogels as well. Enabling tools are propelling this field forward. I've talked a little bit about microtiter plates. Shown here is a spheroid microtiter plate. It's 1536, which means it can go into an ultra-high throughput screening system. Screening 1 million compounds a day and in spheroids is now completely feasible here. And you can start to do sort of imaging here. And this is the imaging of cells as they come together over a period of time, particularly when one looks at them between 24 and 48 hours. And you can start to see that the cells will form a defined spheroid. After 48 hours, you get one with defined edges. Absolutely suitable for high throughput imaging techniques. Just to close the loop on the microfluidics, there are organ-on-chip technologies now entering the 3D culture area. This is one example of an organ-on-chip company with various tissues grown in a microchip environment. The advantage here is that one doesn't just get the microchip environment, one can also have fluid flow across the cell, giving another dimension by which you're mimicking the in vivo situation. And then finally, there is the application of 3D bioprinting. This is one company we've worked with, Organovo, where one can use various systems to print tissues. Here you can print them in 3D such that they mimic the native intestinal model, and then you can start to look at CACO2 as a model of cellular absorption. Just to close then, in terms of applications here, one of the major applications, and there's increasing data published on this, is that novel compounds are being found in drug discovery. Here's an example of one. This happens to use a 1536 Corning plate, and the group from Scripps here identified compounds to alter KRAS mutations, a fundamental mechanism in cancer, and they identified a novel compound that was active that was not identified using two-dimensional culturing conditions. And in addition, here's another application where one is developing cells where one can start to look at glioblastoma, a very aggressive cancer, by looking at cells grown in 3D. You can start to look at the invasion of glioblastoma cells in organoids developed from the patient. In other words, you're starting to mimic, in real time, the implication of cancerous cells in terms of their invasion of brain tissue. And then finally, in the last few slides, there is the area of can you start to have predictive metabolic toxicology. And the US FDA has concluded that liver toxicity is a major issue, particularly in terms of some of the treatments of compounds, for example, with ADHD. And in fact, if you look on this slide, the number of drugs that are withdrawn due to toxicity, where they induce liver and heart failure, it's a leading cause for drug attrition and failure in the clinic. And in fact, the slide here shows that if one studies liver cells in 3 dimensions, not just can you detect compounds that have potential effects on the liver, but you're looking at liver cells that are 3 times more sensitive to potential hepatic toxic compounds. In other words, the ability to predict potential liver toxicity is optimized by looking at liver cells in 3 dimensions, as illustrated here by some data using Corning cells in Corning plates. So in conclusion, the promise of 3D culture is in 3 areas. One of which I've touched on is more efficient drug discovery, but it also holds promise for looking at regenerative medicine, and it also has the ability to speak to the area of personalized medicine, since many cells from individual patients can be grown in 3D, and as a result you can personalize both a disease model as well as the effect of the compound on those patient cells. And my final slide is really a wrap-up slide with some comments here. A very exciting area, a very fast-moving area, an area which is representing years of culmination of cell culture research and providing us with some very powerful tools for both drug discovery and disease research in the future. And with that, I'll conclude, and I'll hand things back over to you, Susie. Thank you.
And thank you, Dr. Eglen, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that this webcast will be available for on-demand viewing through May 2019. As a final reminder, Dr. Eglen will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us today. We'll see you next time. Bye-bye.